Hello everybody, my name is Michal Linial. I'll introduce today something about SARS-CoV-2, and mostly I'll introduce in situ MS proteomics and in view of the integrative modeling. Let's get started. So the outline of what I'm going to tell you about is a little bit about the proteome of SARS-CoV-2. I'll tell you what is the current knowledge on the multi-omics view of this virus. Then I'll introduce the protocol, which we called in situ CLMS, and I'll illustrate how its benefit for understanding the structure and function of 3 different proteins. So this work is done with my friends and colleague at the Hebrew University of Jerusalem, with Dina Schneidman and Nir Kalisman. Now, just to remind us, COVID-19 pandemic in numbers, we know that there are a huge number of cases and with over 4 million deaths, and even now there are more than 12 million currently infected. But just to remind where we have been a year ago in ISMB 2020, in which we had in a completely different state in terms of understanding the disease as of now, and many surprises are still to come. Now, pandemic in general are not that rare. Actually, they are quite common, as you can see here. And COVID-19 has a unique feature that is, if I compare many of the pandemic, including MERS and SARS, that are coronavirus themselves, you can see that the COVID-19 sits in a very stable place in terms of infectivity and mortality, and I think that gives a huge advantage to this virus, and this is all depending on its proteome. So what do we know about its proteome? The proteome builds of 29 proteins that can artificially be divided to those that deals with replication, those that are act in shutting down the cell response, and those that build the virus. Of course, those 30 proteins, we try to understand what they are doing, and we know as of now only partially. But what the protein that we studied very extensively, everybody, is the spike, for a good reason, because it's a cell recognition entity, and it's also the crown of this virus that we can see it from the outside. And what you can see here from a structural point of view, that if we compare SARS from 2002 to that of COVID-19, we see that they bind to the same receptor, but in a different patches, different places. And that's illustrated here, that not only that the binding is in a different place, but actually the COVID-19 and the other viruses of the coronaviruses each have its own way to bind. But for today, the question that we want to focus on is what happened within a human cell. And we won't talk much about the recognition itself. So classically, people in this seminal work of Gordon et al. From 2020, we are able to take each and every one of the protein of SARS-CoV-2, infect to cells, and then do affinity purification to understand which protein binds to this protein, to this viral protein. Indeed, there was a dramatic response. As you can see here in the network, in each red dot is one of the proteins of the virus. A similar work was done in which SARS-CoV-2 was compared with the original SARS from 2002, and this time it was infected into the lung cell, and again, hundreds and thousands of proteins were identified. And this is just a subset of this huge network. As you can see in the numbers, there are over 1,000 viral host interactions that were identified. So the question is, what can one do with such network? And in my view, there are few unsolved questions. For example, are those hundreds of interactions are physiologically valid or relevant, and which interaction are indeed direct. In this study, we seek to understand what is happening in living cell, and we wanted to ask whether we can gain structural information from this study. And to this end, I'll introduce the in-situ CLMS protocol. CLMS protocol works by starting with cloning the protein of interest, here in blue, with a tag in red, infect cell, and then the important part is to introduce the cell with in-situ crosslink that will enter the cell, crosslink the protein, and then and only then we wash the excess of crosslink and release the cell. And from now on, we introduce this affinity purification and mass spectrometry as before. But note that what we are looking are pairs of interaction that are either within the proteins, as illustrated here, or among proteins. So from what I told you is this part of the protocol. From now on, we can use docking experiment, either more classical, which is template-based, or using this crosslink to do integrative modeling and to find the shape of the protein in the cell. So to wrap up this part, I'm telling you that we are trying to probe protein structure inside intact cell in their native environment. Moreover, we are using mass spectrometry to identify pair of protein that are covalently linked and they are in structural proximity. And this is to get a rich resource for modeling for structure and interaction. As you can see, here are the pairs, and here are the crosslink between those pairs. Now, this is the proteome that I already introduced to you, but now let's talk about proteins that are less studied, which start with the NSP1. NSP1 belongs to the replication machinery, which is 16 protein, but surprisingly enough, many of them are unknown, including NSP1. Only recently, there was a very nice work showing that NSP1 actually blocked the mRNA entry point in the 14S of the ribosome. Basically, by its C-terminal, just block the channel, this entry point, as you can see here. And by that, probably shut down the host mRNA on the expense of mRNA of the virus. When we applied our method to this protein, express protein, we found these pairs, and we found it's interacting to a specific protein, which is ribosomal S3 of the 14S of the ribosome. Indeed, you can see this nice agreement with the crosslink in blue, and when you see this, we can dock the protein onto the ribosome and show its interaction to the ribosomal protein S3, and indeed confirm what was shown, that it's blocking the entry of mRNA. So by doing that, we confirm the detailed 3D refinement of NSP1 with the ribosome and explain why this protein indeed block the mRNA entry. Whether this NSP binds to other partner, we looked for enrichment in this binding, and we found only one type of proteins, either the RS3 that I already mentioned and the initiation factor 3. Now, this is in marked difference to the many interaction that was reported by the classical affinity purification, which we couldn't find. The second protein that I want to discuss is a very famous one, is the N protein. And why is it famous? Because it probably helps to pack the RNA within the virus. It is known to have N-terminal and C-terminal domains, which probably binds to RNA or nucleic acid in general. When we looked when we look at maybe just to say that these proteins both have structure already, and it's known to be dimerized in this region. Now, when we look back to the affinity purification experiment that I already mentioned before, you can see this huge network, and we wanted to understand, can we utilize our method approach to assess the relevance of those proteins in this expanded network. And this is those are the results. We can show a very tight interaction here, some interactions that are really long term, and when we map those interactions, those cross-links, we were able to show a very beautiful dimer of two of those proteins, as you can see, the pink and the blue, that are bound here, and we can see that the N-terminal domain is extremely flexible, as can be seen here, and the structural dynamics really satisfied all the cross-links that we observed. We could use this atomic resolution and dock also the RNA, as you can see here in gold. The RNA here are docked, and this docking show that there is indeed a beautiful interface or electrostatic patch that probably fit this V-shaped dimer. So we understand better what is going on with the N protein. And just a last point of interest to this analysis, we were able to show if you take all the variants of the coronavirus that we know of as of today and ask whether there are more or less mutation in the interface versus the non-interface, you see a large difference, and the difference probably tells us that there is an evolutionary constraint on not touching too much the interface in terms of mutation. And we were able really to satisfy all those cross-links, and we were able to show electrostatic that probably driven by the non-specific RNA binding. The last protein that I want to discuss is this NSP, mysterious NSP2, which is again a protein that deals with replication. It has some arresting of normal host cell function with unknown mechanism. Basically, it doesn't have any functional clue. However, it was recently, its structure was solved by the AlphaFold as part of the Cas14 competition, and you can see that it's extremely flexible, and we wanted to understand how does it look in a real cellular system. And in order to do this, I'm showing you here in all those blue lines, the cross-link that really satisfied the locally the subdomain. However, when we go to the AlphaFold model, we see that it has a lot of conflicts. Actually, we had to break down the protein again and reassemble it from scratch. And now, by doing that, 90% of the cross-links are satisfied. So we believe this is what happened in the cell. Actually, we tested directly whether we, in our hand, get the in vitro affinity purification versus the in situ that I explained, whether it's the same. And the answer is not at all. There are many more interactions by the in vitro, and the overlap between the two is only partial. As you can see, in May 2021, a bioRxiv paper with the cryo-EM of NSP was published, in which, again, we do not think this is the right confirmation because it has so many conflicts with what happened within the cell. In order to make sure that we are not fooling ourselves, we tested each and every protein by the antibody to the tagged protein and showed that there is no aggregation in the cells. So basically, our expressed protein are not aggregated and probably show a native state. The last thing that we found is that we were able to find hidden function by this method, and this is a 3 zinc finger or ion binding domain. One of them, site 2, is conserved among other coronaviruses, but neither 1 or 3 are conserved beyond the COVID-19 and SARS-CoV-2. So we believe that this information is really new, and probably NSP2 is a zinc binding protein, as you can see, site 1, 2, and 3, and we were able to find a very conserved region that might be relevant to membrane binding. And why we can say this? Because when you look at the MERS or at the OC43 human coronaviruses or at COVID-19, all of them have a patch, a red patch or a acidic patch that probably binds to lipid or to double membrane. We don't know it yet. The last point is whether we can find the partners of those proteins, and the answer is that the end protein, we could not find any protein binding, despite the fact that it was announced as many binders. We believe that it's the RNA that binds to those proteins, and it's indirect. In terms of NSP, we were able to find few proteins, quite interesting. One of them is ATP shuttling that connect the cells with the mitochondrial function and so on. So to summarize everything, I showed you that we used this cross-linking to capture native structure and protein-protein interaction, and the in vitro affinity purification is prone to non-native interaction. I also showed you that we confirm the blocking of mRNA entry to the ribosome by NSP1. We confirm the dimer formation and the strong RNA binding, and probably driven the condensate state. And on the term of NSP2, we showed that it's a zinc binding protein and much more compact than anticipated, and has a conservation which is a 3D rather than the sequence. And I think I'll stop here, and I'll thank you very much. Thank you.